The magical story of skeletal muscle contraction.
This is a muscle fiber in a muscle cell. This is a bone, and as it connects to the muscle by connective tissue, and the connective tissue comes off from the muscle fibers, just what's coating or around them comes off and forms and extends and forms the connective tissue that connects to the bone. And here's the actual muscle, which is made up of fascicles inside, and the fascicles are made up of myofibers, which are then made up of the filaments, of thick and thin filaments. And within the within the fascicle, there's within the fascicle there's the sarcoplasmic reticulum, which is on the sides of or in between the T tubules that are just an extension into the cell from the sarcomere or sarcoplasm. Alright, this is just an overview of the muscle contraction, and it goes from the beginning with the action potential leading down the synaptic knob to the motor end plate, where in the synaptic knob there's synaptic vesicles releasing the acetylcholine into the motor end plate, where they attach to the acetylcholine receptors, and which then allows the sodium ions to enter into the muscle cell, after which the acetylcholine is attached to acetylcholine esterase, which causes it to break down. Then once the sodium comes into become positive, which causes a depolarization, and it travels down the muscle cell's sarcolemma to the T-tubules and to the sarcoplasmic reticulum. So the calcium is released from the sarcoplasmic reticulum and attaches to the troponin of the thin filaments, which eventually causes the actin to move and expose the active sites on them. And then the thick filaments right here will also move. The myosin heads will attach to the active sites, and once they contract, it'll all contract and cause the muscle fiber to contract. And these are the different muscle fibers, the thin filaments and the thick filaments. And of those fibers, this is going to be called the M line right here. This is going to be the A band, or the H zone. This whole pink thing is going to be the A band, or the whole thick fiber. The I band is going to be right here where it's just the thin fiber, and then the Z disc is going to be right here where the connection of the thin fibers are.
The action potential arrives at the synaptic knob. The calcium channels in the membrane opening, allowing the calcium to move into the cell. The increased calcium causes synaptic vesicles filled with acetylcholine to fuse with the membrane. Acetylcholine is released from the synaptic knob by exocytosis into the synaptic cleft. The acetylcholine is picked up by the receptors on the motor end plate of the skeletal muscle.
Okay.
Acetylcholine is picked up by the receptors on the motor end plate of skeletal muscle. As the acetylcholine is picked up by the receptors on the motor end plate of the skeletal muscle, the sodium-potassium channels open, allowing sodium ions to enter into the muscle fiber. Increase in positive ions inside the muscle cell initiates an action potential on the sarcolemma. The action potential travels down the sarcolemma into T tubules and stimulates the release of calcium, just the little blue dots, from the terminal cisternae, right here, of the sarcoplasmic reticulum, where it has been sequestered by the calsequestrin, the little black triangles, into the sarcoplasm of the muscle cell, which means the calcium will come out down here. And then the calcium floods over the thin filaments and binds to tropanonin. Sorry. Wait,
we can start from
calcium floods over the thin filaments and binds to the troponin, pulling tropomyosin off the active site on the actin. So right there, and the actin is the little yellow.
High energy myosin heads leap to bind to expose the active sites of actin on the thin filaments, forming a cross bridge. Oh, I should do that. Sorry.
Do you want the
High energy myosin heads. Okay.
Okay, here we go.
It's recording. 1, 2, 3.
High energy myosin heads leap to bind the exposed active site of actin on the thin filaments, myosin, forming a cross bridge. The myosin heads pivot, next one, to low energy state, releasing the ADP and moving the thin filament over the thick filament, causing the contraction, the power stroke. ATP then binds to myosin head, causing the head to break free from actin. The myosin heads become energized as ATP is converted to ADP, really energized. Then calcium is pumped, next one. Calcium is pumped back into the sarcoplasmic reticulum and attached to calsequestrin, allowing tropomyosin to once again cover the active sites. There's calcium going into the sarcoplasmic reticulum. And then this is when the actin is, or the tropomyosin is once again covers active sites, resulting in a relaxing muscle. The magical story of skeletal muscle contraction.